everyone. Good to be here. So let me firstly start with a small story I had last week with my son. He came to me, and we've just been to the pharmacy and brought a pack of physiological serum. And he came to me and asked me, "Ma'am, is this product sterilized in the company you are working at?" And I was quite surprised by his question, but also interested. And then we started to look together at the packaging of this physiological serum. And on that packaging, I could find the name of the manufacturer, I could find the ingredients, I could find expiry date, I could also see that it was sterile, but no indication at all about the way it was sterilized and where it was sterilized. So I couldn't answer his question. It's strange, but it's not mentioned on the packaging. And yeah, today I will try to explain you why the method of sterilization is important, not only to say that a product is sterile. Just as a background and to give you a bit of numbers, during the period of 2004 to 2011, FDA made 642 recalls that involving microbiological incidents. And from those recalls, nearly 80% were linked or connected to sterile issues. And part of these 80s, about 80 again, are due to lack of sterility assurance. It hurts. That's really a major issue when you have sterility problems. It hurts the patient due to the lack of availability of the product. It hurts the reputation of the company. It has huge costs, etc., etc. So definitely sterility is a key factor. But what does it mean sterile? So sterile, that's a product that is free of microorganism. Easy to say, not easy to have or to prove. And that's definitely something you can never guarantee totally. So that's probably a unique situation where it's required by law, but you can never demonstrate it. You can never go to 100% of certainty that your product is absolutely sterile. And why? Just because you cannot 100% test it. You cannot do your complete testing of your batch, otherwise it's not sterile anymore. And even if you do a full sterility test, how can you be sure that your sterility test will be able to detect any microorganism? So just for that reason, there is always a finite probability that there are remaining microorganisms on your product, whatever or regardless the extent of the treatment applied. Yeah, so that's why the method you will select for your sterilization, for the sterilization of your product is so important. That's really critical. Let's see together the two possibilities you have to sterilize or to get some sterile drug products. There are really two. The two mains are, first one, that's aseptic assembly. That's a big first way, a big first possibility. The second one, that's terminal sterilization. And as part of terminal sterilization, you will find different technologies like heat, moist or dry heat. You will have radiation and other possibilities like gas with ethylene oxide or nitrogen dioxide, for example. So I propose you to go through these two different possibilities and to compare them a little bit and to see really what are the advantages and of each of them. But first, let's try to see or to try to understand what are really the main differences between those two approaches. So the first one, that's aseptic assembly. And so you see here on the scheme that aseptic assembly, that's really a question of maintaining sterility. So you assemble products or material that are already sterile and you put them together to keep the sterility of your product. Sometimes we can also add microbial reduction step, which is the sterilization. So that's quite frequent in aseptic assembly. And at the end, you get your end sterile product. For terminal sterilization, that's totally different approach, totally different process. There you start from unsterile product, container, closure, or excipients, and so on. And then you do a real sterilization process, and then you get your final product. So there it's really a question of exposure. You expose your product to a physical or a chemical agent to obtain your finished product. And that's why I make the distinction between sterilize, which is more a question of action, doing something, exposing to an agent, compared to sterile. Of course, at the end, I agree that in the two cases, you have a sterile product, but the way to go to that is definitely not the same. And so we will go a bit further and really compare and see what are the advantages and disadvantages of each of these methods. As per the regulation, you cannot really choose which one you want to select. The preferred method, and that's something you really have to keep in mind, that's the terminal sterilization. That's recommended by many regulations in US or in Europe. It's the same. And here I've just picked up European Pharmacopoeia, but you can find it in many other guidance and regulation. And so now we will start to understand why, as per the regulation, as per the authorities, terminal sterilization is the preferred option. So first, with aseptic assembly, I will go through the advantages and through the disadvantages. Of course, that's not an exhaustive list. That's just different main points I would consider for the two methods. Advantages of aseptic assembly. Definitely the impact on the product is quite limited because you just assemble, so you do not have any exposure. For that reason, it's suitable for heat-sensitive products. As you also use different materials, different components, you can have multiple sterilization process for each individual components. Typically, that's an in-house process. And you can use sometimes optimized equipments like wraps or blow-fill-seal technology that gives really good results for this kind of process. And of course, that's a very known and common method in the pharma world. So a lot of companies are very comfortable with that method. For the disadvantages, it's really related to the conditions you have to have for running an aseptic assembly. So really different factors can impact your process, like for example, human factors, also environments. You have to run in a clean room, of course, all the utilities and so on. So any failure that could happen in one of these factors can make your process failing and lead to an unsterile product. Due to all these factors, you have a high level of quality control that is required. And the media field test is one example of this control. You also need to have an additional step, which is usually the filtration. So typically you have that at the end of an aseptic assembly. And it leads to a high-cost manufacturing process. So that's for aseptic assembly. Let's go now for terminal sterilization. So again, the same exercise. So just remember that it's the preferred method, and we will go deeper in that concept. But it's true that it's the validated method. So there you can really validate your assurance of sterility. The process itself, it's made in a standard industrial environment. So you do not need any clean room or any specific conditions to run it. It's an automated process with fixed parameters. So it means that each time you run the same process, you have normally the same result. You can group per product family, or even per category of products. So, and even products do not need to be or to look similar. You can really process together products that are quite different. There is a limited number of quality tests required, and you can treat from really small volumes, like 1 euro pallet or 1 pallet, to a big truck of 32 pallets. So the flexibility is quite large. For the disadvantages, yeah, we talked a bit about that, but that's material compatibility need to be checked. And it will be, of course, correlated to the sterilization method you will choose. Impact on product, efficiencies, but also residuals. Extractable, leachable, as my colleagues talked before, can also be impacted by the terminal sterilization process. You can have higher turn time also, because it's typically a subcontracted process, so you have to go outside to do that. 1 week is a typical turn time for terminal sterilization. Then you have maybe your packaging has to be adapted. If you go for ethylene oxide, you have to have a breathable packaging that's needed. The validation can be complex. You have different parameters to consider. So that's a disadvantage, but it can become an advantage, because you can play on different parameters. Like, for example, you can decrease the temperature and increase the EO concentration. So that's also, it can be an advantage in this case. And you need to do a regular validation or revalidation, but that's the common need for a validated process. But anyway, you have to demonstrate why this possibility, this terminal sterilization, is not possible. And we will go a bit further there, and that's definitely not feasible. So the regulations say demonstrate, but what does this mean really? How far should you go? How far should you demonstrate this? And prior to make your choice or to make your decision, you have to consider really all the different possibilities. It's not enough to say, I go for that option or to that other one. You really have to consider all that is feasible to go for terminal sterilization. Remember that it's the preferred method, so you have to show if it's not working. And there are some mitigation options that are available. So maybe you can adapt your formula to limit degradation, impurities, and so on. Maybe ingredients are not really necessary and could be changed depending on the sterilization method. Maybe you have to adapt your container, or you have to select a component which is compatible with the sterilization process. Maybe also the process itself can be optimized, and then you can limit the impact of your sterilization. So all these things are required. You have to do that. You have to consider these different mitigation options prior to do your selection. And here are some tips or some examples of mitigation for the process itself. For example, in radiation, yeah, you can go for a normal radiation at 25 kilogray, which is the most common approach. And then maybe the output will be my product cannot accommodate that sterilization. But maybe if you try other mitigation examples, maybe it will work. Maybe you can make some inertization of your product. Maybe you can try lower dose. Maybe you can also try to freeze your product and to treat it freeze. So all those examples could help to preserve your product. And maybe at the start it was not, or gamma was not suitable for you, but at the end, if you are able to change a bit or to adapt your process, maybe it will work at the end. And that's the same for ethylene oxide. if you go for a run at 50 degrees at 700 milligrams per liter of ethylene oxide, yeah, maybe your product will not be able to resist to that process. And that's true. But maybe you can optimize your process. Maybe you can just decrease the temperature, decrease the concentration, and maybe to make the exposure time a bit longer. Then it can work. It can accommodate maybe your product. So that's a possibility. Another mitigation possibility, that's to consider a sterility assurance level of 10-3 or 10-4 instead of the usually asked 10 to the -6. That's a possibility, but that one needs to be agreed with your national authority. And there are also alternate methods like an oxylizer, for example, to sterilize your product. So you see that there are various possibilities to consider prior to say my product cannot accommodate terminal sterilization. Yeah, and here really the sequence you have to go for the selection of your terminal sterilization is well described again in the standard. Here I took an EMEA guidance, but you really have to go through a sequential selection process. And the first one is dry, moist heat. That's the first and probably the easiest way of terminal sterilization. But it's not working all the time. If your product is heat sensitive, then you will have to go for another possible method. And the second option to consider for terminal sterilization, that's the radiation. Radiation that can be gamma radiation or E-beam. There are other terminal sterilization possibilities, and we talked about that before, but that's gas sterilization like ethylene oxide or noxylizers. So all these blue ones have to be considered first prior to go to the green ones, which are filtration and aseptic processing, and at the very end, aseptic processing alone. So you really see the sequence of choice. So it's important to use a structured approach to select these most appropriate methods. And that's something we developed in our company, and we call it as a pharma screening, to really make sure that you have good data to support your decision. Maybe it will not work. Maybe you will have as an output that gamma is not working, that ETO is not working, but at the end you have something in your hands to show to the authorities or to your notified bodies or whatever that it's the good choice to go for, let's say, aseptic processing. And then we can go a bit deeper about the effectiveness of these different methods. So we talked about aseptic assembly and about terminal sterilization, but how do we make sure that they are effective? How do we measure that? Typically, in the terminal sterilization, they use the term called SAL, so that's Sterility Assurance Level. So that's more common in the ISO world, I would say, where in the pharmaceutical world and in the aseptic filling, aseptic assembly, that's the PNSU, or probability of non-sterile units. So both of these terms are expressing the efficiency of your sterilization process. But do they mean the same? Are they synonyms? That's something I propose to go through with you now. So for SAL, that's really, the definition is the probability of a single item being non-sterile after being subjected to a sterilization process. So in fact, with a very low SAL, you have a very low likelihood to find an unsterile product. So here, 10 to the -6, which is commonly required for sterile products, mean that you have one chance or one unchance to have an unsterile product on 1 million products. Okay, so that's the meaning of SAL. In other terms, the lowest SAL gives the highest assurance of sterility. And how do we get that? How do we get that number? How do we get that SAL level? In terminal sterilization, it's done through the validation, really a validation process where we anticipate that we can achieve a certain level of sterility assurance level. So here is a graphical representation of the killing kinetic in the during terminal sterilization process. So the pointer seems not working, but I will try to explain anyway. So you see on the there that the population. So here that's a 10 to the 6. So 1 million biological indicators. I will put in blue. That's clearer probably. And you see that there is a direct link between the population on your biological indicator and the extent of treatment. So it can be the time or the dose. So that's really something you can predict. What we do during the validation, we do some testing, we do some experience, where we can demonstrate that at a certain level, you have no microorganisms on your biological indicator. So you are able to demonstrate that your million spores or your million bacteria have been deactivated. So you have a certain time or a certain dose, and then you can double this time or this dose. That's what we call the overkill approach. So really the safety margin we take for that is really high. And so by deducting the level of time or dose, you can show, here you work to the 10 to 0, no microbiological on your BI, on your microbiological indicator. You come from 10 to the 6, then you go for 0, and then you double the time and you go to 10 to the minus 6. And that's how you can demonstrate that you have an SAL of 10 to the minus 6. And this is done with biological indicators. So that's the worst case. That's really a very high population with a very high resistance. Normally, on your product, you have natural bioburden, so which is hopefully lower than 1 million bacteria on it. So you start from, let's say, maximum of 100 CFU on your product, and the killing kinetic is usually really well lower compared to your biological indicators. So really, it means that you have a very large safety margin and a low corresponding risk with such a terminal sterilization process. So that's about the sterility assurance level indicator, I could say. About the probability of non-sterile units. So the definition of that is a probability of one or more microorganism being present on a product item in a population of items. So that's really a detection of a default rate. Then, based on what I explained before, you can easily understand that there is no killing kinetic there. It's really something that you see. You will see a sample that is not sterile. That's how you detect the PNSU factor. And it relies on multiple factors also. So as we said before, the human factor, the environment, the raw material, all these elements will impact your potential PNSU. And how do we estimate the PNSU in a aseptic assembly? That's by doing a media field test, which is corresponding to the worst case situation in your production, and you do that before and after your process. So it means that you can never test really the process itself. It's kind of a bracketing approach. When you say my MFT is okay before, it's okay after, so it means that you have a valid process. But that's how it works in aseptic assembly. So really the most important is to think about sterilization as soon as possible during process development. you understand that's not something you can decide at the very end of the development. The sooner you think about that, the better it is, because if you need to adapt the formula or the container or whatever, that's better to think about that as early as possible during development. And sterility is really much more than just a process. Of course, the process itself, the method you will choose is important, but you also have to consider the whole chain, starting from the raw material, starting from all the different components you will use, also with the partner you will work with, all the individuals that can help you during that process. That's really of the main importance. Here are some examples of products that we have successfully sterilized in our different facilities. So the first one is for gamma, so for radiation, sorry, so gamma and E-beam, so nano suspensions, lyophilized products, dry powder, and API are possible. For ethylene oxide, yeah, we have the sterilization of some APIs. Also pre-filled syringes, that's combination product, but that's something we can sterilize with ETO. We have also some active implants and other examples of auto-injectors, but in this case, for the auto-injector and the pre-filled syringe, that's the external part of the device. So yeah, that's it. Let's guide us to the best method of sterilization, so to the optimal method for your product. So thank you very much. And if you have any question, of course I can answer it now, or you're welcome to our booth to further discuss. You're more than welcome. Thank you.